All right, great. Well, thanks everybody. We can get started. I'm really excited to have Mas come and talk to Cancer Center today about some of his work. So for those of you who don't know Mas, he joined us a few years ago at the Children's Hospital and the Department of Pediatric Hematology Oncology after doing his Hem-Onc fellowship at Johns Hopkins, and he spent a couple years after his fellowship working on actually the topic that you guys will hear about today, and worked really on developing some of the tools and technologies needed to be able to do liquid biopsies and really look at how we can drive pediatric sarcomas forward in terms of therapy. And so I'm really excited for Mas to talk about this work that he's been working on for the last couple of years. And I should also mention that the good news is Mas found out yesterday or the day before yesterday that his DoD grant was funded. So he can continue doing all of this awesome work. So Mas, all yours.
All right, thank you, Rajiv. Thank you everyone for coming to this talk and for the opportunity to talk about some of my work that has been going on for the past few years. So today, let me get back there. So I have no conflict of interest to disclose. So the overall learning objectives for today's talk is to differentiate various liquid biopsy platforms that are currently used and also utilize different liquid biopsy modalities for pediatric sarcomas. So we talk about liquid biopsies a lot, and the term is thrown around a lot, but the main purpose of liquid biopsies is thought to be finding signs of the cancer within the blood of patients. And the things that come out in the blood of the patients that we can see here is things like circulating tumor cells, DNA fragments, microRNA, exosomes. There's a variety of things that you can find. And if you take it in a very wide way of saying it, then probably traditional protein based biomarkers could also be considered liquid biopsies. So how can we do these? So you can use this, you can detect microscopic disease, you can monitor response to therapy. Oops, sorry. You can find new targetable mutations. You can discover new basic biology. So there's a lot of things that you could do from blood-based biomarkers. So for example, detecting microscopic disease. So this was a patient that we looked at circulating tumor DNA. And then while the patient was in radiographic CR, there was still detectable DNA, tumor-derived DNA in this patient. So you could detect microscopic disease before relapse. And you can also monitor response to therapy and all of that stuff. So sarcoma genome as a target is very, as those of you who are not familiar with sarcomas has a wide variety of diagnoses. So when you approach it from the genomic standpoint, the sarcoma genome goes from a very simple karyotype like GIST, chondrosarcomas, translocation-driven sarcomas like synovial sarcomas, myxoid, Ewing, which really don't have, which basically have one or two driver genomic events and then really nothing else, to like the most complex karyotypes like osteosarcomas on the very right, where these multiple chromosomal aberrations happen and large-scale amplifications and loss can happen in their genome. So the easiest target that people thought they could use to do liquid biopsies in sarcomas was to look at these translocation-driven sarcomas. And these translocation-driven sarcomas have very characteristic translocations for each tumor. So for example, alveolar rhabdomyosarcoma, which the name alveolar is starting to a little phase out because that's a morphologic distinction, is driven by a FOXO1 fusion gene. So nowadays, many times clinicians actually call these rhabdomyosarcomas fusion-driven rhabdomyosarcomas or fusion-negative rhabdomyosarcomas. Ewing sarcoma has a characteristic fusion EWS-FLI1, which we'll get into a little bit deeper later. And then synovial sarcomas have SOIT SSX1, SSX2, which is a very characteristic translocation. So the very, very first, and I just love this figure, is from a paper from 1997 that the first thought of using Edavis Fly1 as a liquid biopsy was published. And this is a gel where from lane 3 to 10 have patient blood driven RNA loaded. And then this was an RT-PCR that was loaded on a gel. And you can see that 3, 4, 6, and 7 have positive bands right there at the 270 and 350 base pair expected site. And this was the first proof of concept of finding tumor-derived RNA in blood. So taking this kind of success, there was a large study by COG that was done about in the 2000s, where they looked at finding, I'm not going to get too deep into the flow side, but using flow cytometry to detect circulating tumor cells and also RT-PCR to detect the Edavis-Fly1 transcript. Unfortunately, this study was a negative study, and only 20% of the patients had detectable Edavis-Fly1. And also, as you can see from that survival curve, whether you were able to detect Edavis-Fly1 or not by this RT-PCR really was not predictive of anything. So what turns out with RNA is that mRNA is not stable in peripheral blood, and Edavis-Fly1 is also not an optimal target. So this is just a quick experiment that we did a year or two ago that's right now in revision. But using the left figure shows that using the Streck tube, which is a tube that has RNase inhibitors in it, has significant difference of detection of even housekeeping genes compared to regular EDTA tubes, regardless of when you process it. This was all processed immediately after collection. So there's an immediate degradation that's going on in the blood of RNA. And the right side, we can see that trying to find Edavis-Fly1 and Edavis-ERB fusions in patients, even with RNase inhibition, if you look at the left y-axis, the copy number that you find in the plasma is really, really low. So mRNA probably is not an optimal target. So hopefully we'll stop looking at it. And then, so another version of obviously looking at RNA would be looking at microRNA profiles. And so this is a paper that was published by a Japanese group from their National Cancer Center. And here they determined that looking at 1,000 patients, they found this index, what they call index 6, which is 7 distinct microRNAs, and this was all microarray, that they find that if it is expressed, lead to 90% sensitivity and 95% specificity for the diagnosis of pretty much across the board sarcomas. They compared it to healthy donors, people who have benign tumors, also with a variety of other cancers. And they found that this approach had the strongest data in sarcomas. Now, this is a little bit, I think the criticism of this kind of approach also is that these microRNAs could be expressed anywhere and could be coming from anywhere. So I think it needs a lot more data and validation and real prospective clinical settings to start using these kind of approaches. So since RNA doesn't work, DNA has become very commonly used for liquid biopsies. So ctDNA has been it's been thrown around a lot, and I feel like this is really confusing. So cell-free DNA, circulating tumor DNA, serum circulating tumor DNA, plasma, there's just so many words. So first of all, serum is when you let the blood sit and it's a clear portion left after letting the blood clot. It's now widely recognized that serum has much less sensitivity for circulating tumor DNA. So when you spin plasma and you collect DNA from there, that is defined as circulating cell-free DNA. And that actually includes even exosomes too, because exosomes need specific spinning conditions to differentiate from cell-free floating DNA. So from the circulating cell-free DNA, this includes DNA from the tumor, and then also DNA from normal tissue. And then from the DNA from the tumor, there's mutant DNA, and also wild type DNA is obviously included in tumor driven DNA. So here on the right lower side is a quick figure that shows that there's a peak size of around 150 base pairs, and that's kind of the size that the DNA gets sheared within the bloodstream, and that's the size of the ctDNA that we're trying to capture. And sometimes we've used the word ptDNA because there was an era where people were using serum and plasma interchangeably, and that clearly is not an optimal way to collect ctDNA. So this distinction is quite important because not all cell-free DNA is tumor-derived. So as you can see, this is an experiment we did that I showed a little bit earlier about DNA. And here we show that the copies per ml that's detected for this housekeeping gene, if you just look at the pattern, it kind of looks similar to the right side where we tried to detect EWS-FLI1 and EWS-ERG patient-specific breakpoint DNA. But what you would notice is that these second and third columns, there's 0 EWS-FLI1 or ERG detected right there for this patient, but there still continues to be obviously housekeeping gene detected. And so I think it's interesting that it's also, everything is not tumor derived, obviously, but there seems like more tumor, there will be more cell-free DNA that's floating around, which perhaps makes sense from a cell invasion and inflammatory standpoint. So the ctDNA has really expanded its field in the last 10 years. And this was a seminal paper by Chetan from Hopkins that was on Science Translational Medicine. If you dig deeper, there's like 4 different platforms that he used. But overall, what he tried to show was that across the board, in different platforms, you can really find these mutant fragments of tumor-derived DNA in the blood of really a variety of diagnoses that you can see below. Also, he showed that if you just take it across like all diagnoses and go stage by stage, then the frequency where you can detect ctDNA, and also when you can detect DNA, what the mutant fragment burden is, does increase with, loosely correlates with the staging of the tumor. Now, this is all retrospective, although he had a lot of patients in this. This is all retrospective, and the prospective use of this for disease monitoring is really what the current present studies are doing right now, including our study. So when you look at ctDNA, there's a variety of technologies to assess ctDNA. So there's PCR-based techniques such as digital PCR, which is what our lab does. Its strength is that it's very cheap. Sensitivity is extremely high, and it is patient-specific, which could be a strength or a weakness. And also, so the weakness would be that you can't find new mutations because you would have to run the assay that's specifically targeting an event. Turnaround is very quick because it's just a PCR. You just extract the DNA, load the plate, PCR amplify, and then you're done. You can quantify because it will tell you how many strands of mutant DNA is in there. And then it does require tumor samples often, depending on the setting, because if you really want to know what you're going to look for, then you need to have a panel of targets that you know is present in that patient. So for example, the only FDA approved ctDNA assay currently available uses tumor sequencing followed by ddPCR. And then the more also widely used approach is next-gen sequencing based. So there's gene panels, which you basically find a panel of targets that are known to be commonly mutated in a particular cancer, and then you enrich for that target, and then you sequence them. The turnaround, obviously, because it's a next-gen sequencing based approach, the turnaround is not that quick. And also it is pretty expensive, although next-gen sequencing has become cheaper every year. There's translocation panels. There's one that's currently being used by the COG through Boston, which targets the commonly known translocations in sarcomas. The problem will still be, the challenge will be the same, that it does take time to turn around, and it's still expensive. Whole genome sequencing you could do, and there's varieties of this. There's also ultra low passage whole genome sequencing that people have done, which actually works well when you have a very high tumor burden and high circulating tumor DNA burden. But if you really want to find down to rare mutations, then you really need to sequence deep. So this does definitely become cost prohibitive. So one of the challenges in detecting circulating nucleic acids is that I'm preaching to a choir in this group, but not all clonal events are cancer, and not all somatic mutations are obviously cancer. So this paper in the New England Journal of Medicine was really important to point out that you perform whole exome sequence of DNA in peripheral blood cells, the somatic mutations characteristic of hematologic malignancies were in 10% of patients, persons older than 65 years of age. And obviously not 10% of the patient participants had cancer or developed cancer. So it's something that's really important in this field, that when you're looking for mutant events, you really need to know what you're looking for and what that means for the patient. So in Ewing sarcoma, or in Ewing sarcoma, there has been approaches done by the COG that they tried to look at hybrid capture of the known EWS-FLI1 translocation with next-gen sequencing. So ctDNA detection at diagnosis was around 70% for metastatic patients and 44% for non-metastatic patients. They demonstrated that at diagnosis, if you have a detectable EWS-FLI1 by this trans-seq panel, then your prognosis is not really great. So the challenges of that approach is that, as you saw, even for metastatic patients, you really don't get a good sensitivity or a detection rate. So ddPCR is a now not that new, but relatively new PCR platform that is highly sensitive and specific. And you can use this PCR approach. For example, this was a paper from my mentor, Ben Park, where he showed that detection of PI3 kinase mutations by ddPCR was very successful even after surgery. So post-surgery, 5 out of 10 still had the same mutation detected that were present in the FFPE tissue. So it's quite possible to find these rare events using ddPCR. So we approach this through EWS-FLI1. So EWS-FLI1 is the primary driver, and as I said, an optimal target, and can be an optimal target for circulating nucleic acid detection. So Ewing sarcoma cells always have this type 1 EWS-FLI1 or type 2 EWS-FLI2, and combine these two types, you can see that it basically covers 80% of the patients. So we approached Ewing sarcoma using this information. So in our attempt, we first got the genomic DNA from the tumor, and we made a custom PCR panel that my mentor used to call carpet bombing, that we would just PCR out the breakpoint. And what's interesting is that even though there's type 1 and type 2 EWS-FLI1, the intronic breakpoint of EWS-FLI1 is very patient specific. And I'm sorry, let me, I'm sorry, the Zoom box always shows up where I'm trying to read. So you can design a patient-specific primer-probe combination to target this specific patient-specific intronic breakpoint, and then use that for ddPCR detection. And this approach has been done by our group and also a German group. And it seems like the German group does really well collecting these serial samples over a larger number of patients. But basically, both groups showed that the ctDNA is highly detectable at diagnosis. So around 90% of patients have ctDNA detectable at diagnosis. And the patients who don't recur, so get cured, have quick resolution of ctDNA. So you have negative results and they stay negative. And while patients relapse always have detectable ctDNA prior to the disease recurring. So when we look at, for example, patient number 2 here on our data, there's a surgical resection and that led to radiographic CR. Later, the patient does relapse, and you can see that it was already, the ctDNA was detectable there. In the Krumpholz paper too, what's notable, for example, this fourth right lower patient, the disease burden detected by radiographic evidence comes up way later, 75 months after diagnosis, while the ctDNA burden was detectable even 6 months before that was happening. So even though there was concurrent imaging that was happening. So this shows the strength of this approach and finding MRD. So as I said earlier, fusion genes are common in sarcomas, and there's definitely potential for expansion. We do have, we've started looking at alveolar or fusion-driven rhabdomyosarcoma and synovial sarcoma about 2 years ago, and we've collected some patients' serial blood samples and have been able to detect these fusion genes. So taking a little different turn. So that was kind of the easier, simple side of the genome of sarcomas and Ewing sarcoma and translocation-driven sarcomas. Next, I'd like to talk about osteosarcomas, one of the most complex chromosomally rearranged sarcomas. So this is a paper from Alejandro Sweet Cordero's group from UCSF that was published 2 years ago, and it's considered somewhat a seminal paper in this field, where until then everyone was trying to find mutations, and there are definitely recurrent mutations like P53 and RET, but also he really showed that you can subgroup these patients based on these recurrent somatic copy number alterations. There are recurrent events that happen, and you can really group patients based on what kind of recurrent alterations or large amplifications happen. So if you take that approach to osteosarcoma, left side is what's published from what was published 2 years ago from Brian Crompton's group at Boston. You can see that using this ultra low passage whole genome sequencing, it's the I-core-CNA from the Broad, you can see that there's these large chromosomal gains and losses that you can detect in the blood, which is a lower box, which follows exactly what they found in the tumor. So the right side, this is unpublished data, but you could also find in a serial sample that post-surgery, so when you were supposed to be theoretically CR, you really don't find anything even in ultra low passage. 3 months after surgery, you start finding these chromosomal rearrangements, so copy number alterations. And then 5 months after surgery, when the patient is clinically progressing, even in ultra low passage, you can actually find a lot of tumor DNA just by this ultra low passage. So detection of these changes at diagnosis definitely can predict your outcome. If you have ctDNA detected by ultra low passage, then you do worse in your outcome. Although, I mean, one can argue that whether this kind of difference is really that relevant clinically. And also the problem with osteosarcoma right now is that even if you know you're going to do terrible, there's nothing you could change with the therapy right now. So another approach, because this is just ultra low passage, is to use ultra deep targeted sequencing. So as I said, even though this osteosarcoma is a copy number driven disease, you can, there are recurrent mutations. So you could put together a well-designed targeted sequencing approach for osteosarcoma covering these commonly known mutations. So this is data from our close collaborator, Dan Weiser's group, that we pretty much share data and assays across our groups, where he shows that in a patient where there was initially surgical removal and radiographic CR right there, you do start to see this spike in ctDNA after the patient relapses. And then also after resection of the Mets, there is a drop, but still present circulating tumor DNA in this patient. The right side is a little bit busy diagram, but what it shows is that the boxes, so these are A, B, C, D, E, F, G, H are all patients. And then the boxes are where the patient had detectable ctDNA by this ultra deep targeted sequencing approach, where you could see that many of the patients who have detectable ctDNA events happen. So the current and future directions of these approaches that we really need to, there's, including our group, there's multiple pilot studies that have been done. The platforms are there. The assays are pretty much automatically designed. So we really need a larger prospective study to validate the use as an MRD marker. So I have a study going on across 20 institutes that in a group called NPCF, where we prospectively are collecting plasma and also analyzing these already developed ctDNA assays to see if it can predict relapse. So to really prove it for the use as a MRD detection biomarker. The study has been open for almost a year and a little bit, and due to COVID, every center pretty much closed enrollment, including ours, closed enrollment to the study because it's considered a non-therapeutic study. We've gradually started to reopen. So there has been a very unfortunate delay with accrual, but we've really started catching up accrual this year. Also, the COG, which is the National Pediatric Oncology Group, is including plasma collection in the currently planned therapeutic protocols that are out there. And we're part of the osteosarcoma collection protocol, and they will send samples to us. And also, there's a Ewing trial that's currently planned, and we are in the running to get samples from that trial too. Other future directions for ctDNA is also figuring out what's going on with the clonal evolution with sarcomas. The unfortunate thing with sarcoma research is that samples are very, very hard to come by, and there's a lack of metastatic and recurrent patient tumor samples. And so this is right now viewed and positioned as a way to look for treatment-resistant mechanism of treatment resistance and clonal evolution in sarcomas. And really, right now, the current limitation is not necessarily the technology or the science, but more of lack of well-annotated samples. So taking a little different step, I'm going to talk about a little bit about circulating tumor cells, which is another large portion of our lab investigation. So as we all know, metastasis is very complicated, and circulating tumor cells need a lot of things to overcome to metastasize. The circulation itself is not a natural environment for circulating tumor cells, and it is said that less than 1% of tumors that are shedded into the blood actually can survive, which somewhat makes sense when you think that people can't survive if they just fall off the Niagara Falls. So there have been many ways that have been used to detect circulating tumor cells and in carcinomas as a more common cancer. There have been the commonly known cell search system, which uses EpCAM as a positive selection mechanism. There has been negative selection, just taking out all the CD45 positive cells from blood and then using, for example, flow cytometry or other capturing mechanism after that. Physical properties have been used and have been successful, such as size-based filtration or FICO just gets a mononuclear, but then after that, excluding the cells. DEP arrays have been used. There's the single spiral microchannel is mostly has been used in a Boston group that uses a vortex that finds size-based cells. So there are different platforms that have been used over the past decade, but the translation to sarcomas have been quite challenging. So these are a couple of previous efforts that were used. Probably the largest study that was used was this middle clinical cancer research paper. So this was based on a paper from on pediatric blood and cancer in 2010, where Steve Du Bois showed that in one patient, he could find that there were more CD99 positive cells in the bone marrow than healthy patients. CD99 is widely used, especially in diagnosis, to diagnose Ewing sarcoma and can differentiate Ewing sarcoma from other blue round cell tumors. However, CD99 is positive in primitive lymphocytes, and it's now well known that just looking for CD99, you're capturing a lot of other cells. So this was taken to a national COG study where they ran flow on basically all the diagnostic bone marrow and peripheral blood samples, where they found that finding these cells really can't make a diagnosis or and also couldn't predict much. And then other platforms that were used was on the top is universal mark. So this is a paper by MD Anderson that they found that cell surface vimentin, which is a antibody that they in-house developed, could differentiate sarcomas from other blood cells. So this is the challenge, is that sarcomas aren't carcinomas and we can't share the same platform. So we can't use same markers like EpCAM to find CTCs. So our approach has been using size-based selection with this cell-sieve filter system, and it has a 7-micron pore and directly can process peripheral blood without much pre-processing. So this can be adapted for sarcomas. It was initially published in carcinomas, but we quickly found that we could adapt this for sarcomas using the vimentin positivity, CD45 negativity, and find these giant cells that are floating in the blood. And these CTCs and CTC clusters can also be detected from mice. And you could see here that this is our amputation mouse model, but if you amputate the mouse right here, and you should only have micrometastatic disease, you actually find that post-op day 9, the CTC numbers come down nicely. And then when they start having metastatic relapse or recurrences, their CTC number significantly goes up. And these CTC and CTC clusters of sarcomas can be detected across multiple sarcoma types. And we've shown in few papers that it can be found in osteosarcomas, Ewing sarcomas, MPNSTs, alveolar rhabdomyosarcomas, and that we can, if in a controlled setting, we can often use this as to predict future relapse. This is a non-transplant patients, and in transplant patients, the data would seem to be a little bit less weak. There were patients who had persistent CTC who had longer survival, which could be related to maybe immune surveillance, but we don't know much about that setting. So although these CTC is, I think, is a really cool platform for as a biomarker, the challenge has been that the number that you can collect is really not that high. So there's only a few cells that you can collect even in metastatic patients. So then now our newer approach has been to try to better understand metastasis biology through looking at CTC collected through cell sieve. So what we've been doing is we take the cell sieve membrane, enrich for circulating tumor cells from patient blood, and then use 10x single cell RNA-seq to sequence these enriched samples in order to differentiate the circulating tumor cells from the background cells. So for example, in osteosarcoma, which is a significantly different cell type than blood, the left side is a patient biopsy where we show that from the biopsy, there's the tumor cells that are highly spark and CALD1, which are both bone-driven proteins. So they're both highly expressing spark and CALD1 on single cell RNA-seq. And then when we take that profile into the enriched samples, well, we do have a large number of white blood cells that are contaminating the sample, which is by design. We are able to find this small cluster where there's an enriched CTC right there that is single cell RNA sequenced and express spark and CALD1. These cells compared to the tumor types, just this was a quick IPA analysis that we performed, is enriched for signaling such as integrin signaling, leukocyte extravasation signaling, IL-8, which is known to be a strong driver of osteosarcoma metastasis, actin cytoskeleton signaling, which makes sense for a cell that's floating in the blood. And then Richter was one of the highest Z-score of predicted upstream regulators. So we could definitely look at the biology of what's going on in these circulating tumor cells compared to the primary tumors. And there's still ongoing collection locally for these kind of data sets. And also we're right now combining this approach into the 20-center multi-center study where we're also collecting blood, circulating tumor cells, and also local tumors as a more larger scale study. So the current future directions for CTCs for sarcomas really is trying to figure out, we have the technology, we can find these cells. Are CTC a biomarker of metastasis, recurrence, disease burden? The current understanding seems to be that it might be more about risk factor of metastasis and recurrence, but that really can't be done unless you have a larger scale study, which we are currently doing right now. And then also, as I mentioned in the previous slide, how are we going to discover, what can we discover of how tumors evolve from a local tumor to shed into the circulation and survive as CTC, which could be a very powerful metastatic, anti-metastasis target. So thank you everyone for listening. I just want to acknowledge all of our great patients who really helped us in this, a lot of these studies. And then our lab, the solid tumor group at Children's, which really has helped me in collecting these samples, amazing collaborators and really great mentors who have been supportive of me through these past few years as I transition here, and then some of our current funding sources. Thank you. Any questions?
There are some questions on chat, just so you know. But one question I have: well, how would you approach a amplification based, such as a dedifferentiated liposarcoma patient?
So I think the easiest approach would do ultra low passage, looking for the amplification and the copy number, just doing a quick copy number analysis of circulating tumor DNA. Or if we do, I mean, that would probably be the cheapest way to do it. We could do straight up whole genome sequencing on the genome because we can differentiate the copy number aberrations. So that probably would be the approach I would take.
I'll leave the talk to you. Nice talk.
Thank you. I see Jean's question for after. How do these technologies compare to what they use for MRD? Great question. So we're trying to, so one of the primary aims of the multi-center study is to see if the drop of ctDNA from diagnosis to cycle 2 can be predictive of patient response that we could find out later during surgery from percent necrosis. So trying to position it or try to take the MRD approach that's used in leukemia. For the sensitivity and specificity compared to leukemia, that's very tough. The question that has been really around ctDNA is how you define sensitivity. So there have been papers and figures that have come out trying to say that you can find 0.1%, 0.01%. But as I showed earlier in the talk, cancer patients have more cell-free DNA in general. So what does it mean if you have a tremendous amount of normal cell-free DNA floating around and you just find 0.001% of that as tumor driven? Does that mean you have a 0.001% sensitivity? I don't think that's not the case. I think what's clinically more important is how sensitive is this to find out if the patient has MRD in fact, as in, I think, shown by later relapse. So that is definitely, we powered our multi-center study to be able to detect that, as in detection of ctDNA at time of CR and how predictive that is of future relapse. And then does one need to know the difference between exosome DNA and true cell-free DNA? Yes. That's an excellent question. This is something that was raised in our review of a recent paper. So currently, my understanding of the difference is that exosomal DNA and RNA is still contaminated within the cell-free DNA that we collect. So when we do simple cell-free DNA collection, we just spin it down at a centrifugation rate that is much lower than what's used for exosome purification. So there's significant contamination of exosomal DNA and RNA within what we collect. I think, so practically saying that's probably not a problem, because what we're trying to do is find whether, trying to find if you can find tumor-driven DNA or RNA. So I don't think there's, clinically it makes a difference whether we find it from the exosome or cell-free DNA. But if it is from a biological standpoint, obviously I think exosomes and how cells communicate with each other are really interesting. There has actually been a study trying to look at EDOVS-FLY1 fusion RNA within exosomes, and it turns out the fusion transcript, which actually is a transcription factor, is not secreted within exosomes. So I think there's a lot we need to know more about the biology of it, but as a clinical biomarker, it doesn't seem like it makes a difference. And then Anthony. Yeah, so the Natera approach, I think, is great question. So Natera approach is very similar to pretty much what everyone has been doing, that they sequence tumors and then they make a ddPCR panel and then they just run ddPCR in the blood. So I think their strength has been their ability to commercialize it and get it FDA approved for a process that pretty much like anyone does. So I think their approach is solid and their sensitivity should probably be very good. And they should be getting, Anthony, does Natera return results and positive, negative, or quantification?
You know, I'm not 100% sure. I think they do quantify. You know, one of the issues with cell search was we all know how to collect CTCs and or ctDNA to a certain extent. Obviously, there are technical challenges. But one of the things that was seen as clinically useful is to be able to measure trends.
Yes.
And, CellCearch took the position, okay, we're not going to do it per normalize for amount of DNA. We're going to just normalize per volume blood.
Yes.
And I think that was brilliant because that has actually less bias probably and would potentially, decrease the problem that you just mentioned about increased circulating DNA in cancer patients in general.
Yeah.
Because it would give you a different way to normalize.
Agree.
So they do provide sort of changes over time. But
Yeah.
I don't remember quite how they normalize it.
Sure. So for example, for what our lab is doing, we're actually only reporting mutant copies per ml of blood as a quantification, because our platform specifically detects the mutant DNA. So it's probably going to be reported in the same way that CellSearch did. The Natera approach, I think, should be able to quantify because that's what we do. I don't know how they're reporting it. My guess would be that they either report it by fractional abundance, which is probably a carryover from the next-gen sequencing days, but still people use, but it's not as accurate as you mentioned because of the background. But if they're reporting just the mutant events, I think that would be a pretty reliable way to report it. All right, any other questions? All right, I think